Nature is biochemically chaotic. A plant growing in the wild absorbs different nutrients, faces variable climates, and develops an unpredictable matrix of compounds. Modern pharmacology, however, demands absolute precision and sterile consistency. According to the World Health Organization, 80% of the population in developing countries relies on traditional plant-based medicines. India and China alone hold 40% of global biodiversity, serving as the primary producers. But you cannot simply pull a root from the soil and hand it to a patient in a modern clinical setting. That raw biological material must cross multiple technological thresholds to become a regulated, safe therapeutic drug. This is the entire purpose of herbal drug technology. It is a strict engineering pipeline designed to strip away botanical variability, isolate the active chemistry, and extract consistent medicinal value from organic chaos. Regulators use a strict definition cascade. At the base is an herb, crude plant material. Next are herbal materials, like natural extracts or oils. Processed, these become herbal drug preparations. Finally, finished herbal products are clinical outputs, combined with excipients and ready for patients. Identifying exactly which of these 4 stages a material occupies dictates every subsequent processing parameter, chemical testing method, and safety protocol applied to it. The pipeline begins at the source. Herbs traditionally gathered from a wild source are economical, but you cannot predict quality or yield. Therefore, modern manufacturing favors the cultivated source. Systematic crop planning ensures a reliable yield, consistent purity, and regular supply. Market adulteration triggers a mandatory authentication sequence. As seen here, regulatory standards require combining multiple procedures, evaluating the plant's macroscopic and microscopic characters, running chromatographic procedures, and testing specific chemical reactions. When basic morphological or histological characteristics fail to verify the drug, pharmacologists deploy advanced DNA methods and complex chemical assays to prove the exact genetic identity. Using the wrong drug is ineffective and can actively worsen a patient's condition. Rigorous sourcing and multi-tiered authentication act as the critical first firewall in the manufacturing process. Once authenticated, the plant enters primary processing. As outlined in this flowchart, this is a sequence of physical interventions designed to strip away dirt, foreign objects, and excess moisture. The first mechanical step is garbling or sorting. This strips out organic waste like excessive stems, stalks, roots, sand, and dirt. This process utilizes specialized industrial equipment. When processing castor seeds, powerful magnets are passed over the raw material to remove stray pieces of iron. Next is leaching, where the plant material is subjected to running water to wash out impurities. Crucially, operators must strictly control the duration to prevent loss of chemical constituents. The most critical primary step is drying. Removing moisture immediately halts microbial and mold infestation. Crucially, it prevents the activation of internal plant enzymes that would otherwise degrade the active ingredients. Thermodynamics play a huge role. Natural sun drying works for robust materials like seeds, but plants rich in volatile oils must be shade-drugged. For industrial-scale efficiency, artificial drying is utilized. Temperature-sensitive drugs require vacuum dryers, while moisture-sensitive compounds use spray dryers. Secondary processing deliberately alters the plant's chemistry to improve safety or efficacy. Aging cascara bark for a year reduces severe irritants. Alternatively, sweating herbs in high humidity at 45 to 65 degrees Celsius induces hydrolytic and oxidative reactions that break down specific ingredients. The core of herbal drug technology is extraction. This diagram illustrates chemical separation using selective solvents. The targeted liquid solvent is called the menstruum. The depleted solid waste left behind is the mark. There are several ways to execute this. An infusion extracts materials using cold or hot water without boiling. A decoction strictly requires boiling the herbal materials with water to break down tougher cell walls. This extraction phase represents the critical pivot point. The complex, fibrous botany is left behind in the mark, and what remains in the menstruum is pure, concentrated pharmacology. We can see how this phytochemistry operates in practice by looking at everyday plants. Take garlic, or Allium sativum. Cultivated globally, its bulb contains an unusually high concentration of sulfur. This reaction equation illustrates how crushing a garlic clove releases the enzyme alliinase, which immediately targets a compound called alliin. This reaction creates allicin, imparting the pungent odor and acting as the direct source of garlic's pharmacological activities. Or consider chicory, widely cultivated alongside other crops in Pakistan. Its pharmacological value relies on specific constituents: chicoic acid, obtained from methanolic extracts, and the anthocyanins responsible for the blue color of its perianth. A plant's therapeutic power is mechanistic. It is bound entirely to these specific, isolitable chemical pathways. Here, we encounter the primary clinical danger of herbal medicine. Conventional modern drugs typically deliver a single, highly refined active ingredient. Herbal medicines deliver a vast matrix of dozens of pharmacologically active ingredients simultaneously. Because of this multi-molecule payload, dangerous herb-drug interactions are statistically more common. This is exceptionally risky for conventional drugs with a narrow therapeutic index, like warfarin, insulin, aspirin, and digoxin, which have a tiny window before a toxic overdose. Introducing a complex swarm of herbal compounds into a patient's bloodstream can unpredictably alter how these narrow-index drugs are absorbed or cleared by the liver and kidneys. The chemical complexity that gives an herb its traditional healing power makes it highly volatile when mixed with modern synthetic pharmacology. We see this clearly with St. John's wort. It effectively treats mild depression, but its chemical matrix dramatically speeds up the clearance of life-saving drugs, rendering them ineffective. Furthermore, large oral doses alone are highly unsafe, triggering severe skin reactions. Other supplements carry severe hemorrhagic risks. Salvia and ginkgo strongly enhance anticoagulation, causing dangerous bleeding if a patient takes standard blood thinners like warfarin. Additionally, ephedra's interaction potential is so severe that concomitant use with caffeine has resulted in fatalities. Natural does not inherently mean safe. These are powerful chemical cocktails that demand the exact same clinical respect, scrutiny, and management as any synthetic prescription. From harvesting and drying a wild plant to the targeted chemical extraction of its menstruum, every step in herbal drug technology is a deliberate engineering choice. The objective is to capture the therapeutic benefits of nature's biochemical complexity while standardizing it to prevent clinical chaos. As global reliance on traditional medicine expands, we cannot rely on raw roots and leaves alone. Applying rigorous, cutting-edge technology to ancient botanicals remains the only verifiable path to guarantee both clinical efficacy and absolute patient safety.